Okay, so you were born with a set number of muscle cells and neurons, nerve cells, from your brain to your spinal cord. That's the number of the cells you were born with, and that's the number of the cells you will die. Same as the muscle cells in your body. Skeletal muscle, smooth muscle, those numbers are set, whatever it is, 10 million, 20 million, all these years. I should have researched it and found out the number, but what's the point? I don't expect you to know the exam. But 10 million muscle cells, you will die with 10 million muscle cells. Am I making some sense? Unless some kind of disease happens, some kind of cancer happens, the muscle cells increase, or some kind of disease occurred, the muscle cells undergo atrophy, they die. That's abnormal. I'm talking about normal cases. You are born with a set number of muscle cells and neurons, and you die with those numbers. That concept in biology is called eutelly, E-U-T-L-E-Y, eutelly. And I already wrote it on a report last time for you guys. So what happens in case of pregnant women or Arnold Schwarzenegger or whoever is bodybuilding, the number of the cells do not increase. You all know what I'm talking about? Pregnant women, the number of the cells do not increase. It's the size of the cells that is increasing. Stephen, am I making some sense? Okay. So after pregnancy, those muscle cells must go back to their normal size. And what happens when the muscle cells are going back to normal size, lysosome release some enzymes to put back those cells to the normal size. It digests away extra protein molecules that are within the cells, and the cells will go back to its normal size. Samir.
So when a cell dies, a new one forms? Because you said when the lysosome releases all of its enzymes, the cell dies.
In severe cases, right here, what happens in the case of rheumatoid arthritis, the nickname for lysosome is called suicide cell, suicide bag of the cell. If this breaks open right here, this breaks open, what will happen, all of those enzymes, it will kill the cell. That's called suicide bag of the cell. But this lysosome release some of its enzymes one at a time, slowly, not all of them. If you release all of them, then the cell will die. But if you release just a few, then the cell is supposed to release a few of them.
Yeah.
Okay? That's it. That's all I'm trying to say. Did I answer your question?
Yes.
Okay. So, and in case of rheumatoid arthritis, they say the lysosome is malfunctioning and releasing a lot of enzymes. The other one, when our white blood cells, this is a white blood cell right here, eats up, phagocytize bacteria. And when bacteria gets inside of the cell, then lysosome release its enzymes and break down those bacteria. So the bacteria cannot remain inside of the cell. They have to die. And that's another function of lysosome. Lysosome fuse with food vacuoles and digest the molecules. Atrophy is autophagy. It means recycle cell old organelles. You saw it in the video that this mitochondria, what will happen, this mitochondria, after a while it gets old. Okay, when it gets old, then it has to die. What will happen, lysosome release its enzymes and it will kill this mitochondria. Okay, so those are some of the functions of lysosome. And that process is called auto, it means self, based on your prefixes, and suffix auto, it means self. Autophagy, phagy means eat. Phagocytosis, cell eating. Okay, here are some pictures again showing you how was digestive enzymes in lysosome attached to the food vacuoles, and then of course they will break down whatever the material is inside of the food vacuoles, digest them away. And same as this one is autophagy. This is phagocytosis, cell eating, and this is autophagy when an organelle, like a mitochondria, is getting old, of course, a lysosome attached to it and then breaking apart. Okay, vacuoles. Let's talk about vacuoles, and then we tackle that one up today a little bit. What are vacuoles? Diverse maintenance compartments. Vacuoles are large vesicles derived from the endoplasmic and Golgi apparatus. Vacuoles perform a variety of functions in different kinds of cells: food vacuoles, contractile vacuoles in protista, and central vacuoles in plants. What do you think the biggest organelle here is in this plant? Huh? A vacuole. Vacuole is the biggest. Look at it. The biggest structure in here is a vacuole. Right here. So in plant cells, vacuoles are the biggest organelle. And did I define organelle for you guys or no? Did I? Organelle is a membrane-bounded structure, a membrane, so vacuole has a membrane. Is a membrane-bounded structure that has a metabolic function. That's the definition of an organelle. Okay? So vacuoles are large vesicles derived from, they came from, endoplasmic reticulum and Golgi apparatus. And vacuoles perform a variety of functions, and food vacuoles. Contractile vacuole in protista, and I will talk about the other one. Here is a contractile vacuole. You will do an experiment with diffusion and osmosis today. A lot of water comes inside of this amoeba. This is a model of amoeba. So when a lot of water is coming inside of amoeba, what will happen if this cell does not have a system to get rid of the excess water, what will happen to this cell, you think? Huh? It will explode. It will explode. Too much water inside, it will explode. Okay, so it has a system. This is called contractile vacuole, and it's attached to the cell membrane. You see it in the back, Maya. It's attached to the back, and then the excess water goes inside of the contractile vacuole and get rid of, and then squirt itself, so the excess water will get out of the cell. So the cell will survive. The excess water that goes inside of it. So that's what a contractile vacuole is. And in plant cells, of course, it stores food, nutrient, right here. That's what is stored in there. Food vacuoles are formed by phagocytosis, and a contractile vacuole is found in many freshwater protista pump excess water. I talked about that. Central vacuole found in many mature plant cells hold organic compounds. Here they are transmission electron picture of a central vacuole right here. Okay, so you might see these, and I will point out what is the name of structure right here, and then you should be able to identify. Endomembrane system, a review quickly. Endomembrane system is a complex of dynamic players in cell compartmental organization. And here they are. Here's a rough endoplasmic reticulum, make a vesicle. Inside of that vesicle, possibly there are protein molecules. That membrane outside of this, that membrane with particles inside of it, that membrane fuses with Golgi apparatus membrane. Inside of Golgi apparatus, you add carbohydrates to it if it needs it. Not every protein has carbohydrates attached to it. And then what happens, that carbohydrates attached to it folded, Golgi apparatus folded, and then possibly they send that vesicle out throughout the body. The Golgi apparatus, you learn that term cis and trans. Remember cis, it means they're both on the, what was it that we talked about, fatty acids, when both functional groups are on the same side. If you remember that stuff from exam number 1. And then trans, it means they're on opposite side. Here, really does not mean that. A cis side of Golgi apparatus, it means it is close to the, more inside of the cell where the endoplasmic reticulum is. A trans side, it means they are going to transfer their products outside of the cell. So based on this model, if you look at this model, the cis side of Golgi apparatus would be right here. That would be the cis side of it. A trans side of it is closer to the cell membrane. I'm going to make it some sense what is the cis and trans side of Golgi apparatus. Another name you should know, I'm sorry I did not mention that before. Maybe I did mention it, but I don't remember. As the name of this molecule, it's called glyco. What is glyco? Glyco protein. What does that mean? Huh? Yes.
Proteins covalently bonded
To what?
Carbohydrate.
Very good. So right here, you have protein coming from endoplasmic reticulum, and in Golgi apparatus, when you add carbohydrates, Golgi apparatus, covalently, she said, add to the protein molecule coming from here, then the name of that molecule is not protein anymore. It's called glycoprotein. Do we have, I will come back to it. We do have also glycolipid. What is that? Same thing, but attached to glucose. Carbohydrates are attached to lipid, and you will see that. Okay, what is mitochondria and chloroplast change energy from one to another form? Mitochondria, chloroplast, peroxisome. Mitochondria, as you mentioned, cellular respiration occurs in there, makes a lot of ATP. We will talk about it in more detail. There's one entire chapter on mitochondria of your textbook. There is one entire chapter in this exam number 2, is on chloroplast. Now go have fun with it. Maya, when you go home today, on a dinner table, it says, I'll learn about mitochondria and chloroplast. And they probably beat you up on the dinner table. So what are you cussing at us? Say, no, mitochondria gives you energy. Chloroplast, without it, we cannot live. Right? Something like that. You try to convince them so you can eat more on the dinner table. Peroxisome are oxidative organelles. What is the evolutionary origin of mitochondria and chloroplast? And the name of I talked about it, it's called endosymbiosis. Endosymbiosis. Symbiosis, it means living together. And then endo, it means inside. So these two organelles were possibly evolution. We are talking about evolution in old days, millions of years ago. They were bacteria, and these cells engulfed them. This one does not do photosynthesis. This cell engulfed. You all know what I mean, engulfed. ATE, not AT&T. They're no good. I mean, they're good. I have AT&T, but I don't trust them too much.
Same.
Right. I'm being on YouTube, and imagine what if they go after me. I said, ATE, ate. They ate bacteria, and that bacteria over a period of time. The story is a little bit longer than this. I'm just giving you a short one. Here they are. Here is a picture of endosymbiosis. Marisa, is that right? Maricela. Wow. How am I going to learn all of that? And I have until May. Marisa, Maricela, and Maya. Oh God. What else do we have starting in M? You guys, you know what you guys remind me of? Michigan? What is the abbreviation for Michigan? Anybody knows? MI? I guess.
No.
What is it?
I can't remember my notes.
I can't remember. What is the abbreviation for Minnesota? MN. Very good. What is the abbreviation for Mississippi?
Oh, Michigan's MG, isn't it?
MG? I don't know.
MI?
MI? What is for Missouri then? What is for Minnesota? What is for Mississippi? What is for Missouri? What is for Massachusetts? That's what these guys remind me. Maricela, Marisa, Maya, Mendoza. Oh boy. this semester I'm gonna have fun. Trunk, I like you. Because you're just Trunk. You're not Melissa or Maya or it's gonna be hard this semester. Man, it's gonna be man here, man.
That's his
Another
last name,
M.
man.
I know it's his last name. Mendoza is the last name too.
My last name is Mansour.
Has M also?
Yeah, Mansour.
Mansour, oh boy. Oh boy. Oh boy. I'm in for a treat this semester. I will learn your names. I will. Non-V. Two N's. Non. N-A-N-V-E-E-T.
NAV.
Huh?
NAV.
Oh, there is no two N's then. Navit.
Yeah. NAV is an N-E-T.
Okay.
Yes,
Oh.
two N's. Two N's.
Two N's?
Yes.
Non-Vit.
Yeah.
Yeah, I said it correctly first time.
Yeah. Now you're right.
Yeah. Another thing about mitochondria and a few other structures, they have their own DNA, they do multiply on their own, they have chlorophyll, perform photosynthesis, chloroplast perform photosynthesis, is that right? Okay, and then plants, algae, fungi, protista, and some bacteria. What am I saying? What is anatomy of mitochondria? Chemical energy conversions. Okay, what am I saying? Plants, algae, and some protista, they do photosynthesis. Okay, that's what plants, algae, some protista, and some bacteria perform photosynthesis. Okay, that's what. As chlorophylls, we'll talk about that. What is chloroplast? Capture of light energy, and they have their own DNA. These two organelles, beside nucleus, these two organelles. So there are three organelles inside of a cell that have DNA. Nucleus, of course, the brain of the cell. Mitochondria, the power plant of the cell, and chloroplast. They have DNA. They have their own DNA. Golgi apparatus, does it have DNA? Huh? No. No. Endoplasmic reticulum. Smooth, rough. They do not have DNA. Okay. Here they are, electron transmission and picture of mitochondria, and an electron transmission of chloroplast, and we'll talk about that. What is peroxisomes? They do oxidation. Peroxisomes are specialized metabolic compartments bounded by single membrane. Peroxisomes produce hydrogen peroxide, H2O2, and convert it to water. That's what the function of it is. How peroxisomes are related to other organelles, still don't know. Maybe you get your PhD in it, Trump. Whenever you might get your PhD in it and work out the function of peroxisome. Okay, if you're interested in peroxisome. Mendoza, right? Here they are, chloroplast, peroxisome, and mitochondria. Okay, the cytoskeletons, let's get in there. The cytoskeletons anchoring many organelles. There are three types. Your textbook gives you the number. Do not worry about the number, but I'm worried. You should worry which one is the thickest, which one is a middle part, thinnest, and which one is intermediate. And the thickest one has a name called microtubules. Microtubules is the thickest one. And then microfilament, easy name, micro, it means smallest one, the thinnest one, thinnest. They're like a tube. Inside of the cell, they're like a tube. And outside of the cell, of course. They can be found outside of the cell. Intermediate filaments, they are middle size. So they are both are inside and outside of the cell. Support the cells and maintain this shape. That's the function. And then it interacts with motor proteins to produce motility, and I will talk about that a little bit later on, maybe today if the pictures come up. If not, we'll talk about it. Inside the cell, vesicles can travel along tracks provided by cytoskeleton. So he did talk about it. Might as well. Let's talk about it. Right here, can I erase this?
Yeah.
Because I want to be in the video. Hopefully most of this is in the video. So what does that sentence mean? It means inside of a cell You guys cannot see anymore. But that's it. Inside of the cell, the last sentence. I'm going over the last sentence. Okay. What it is that you have a cell right here is a cell, and inside of the cell you have, of course, you have nucleus, you have endoplasmic reticulum, right? And then you have tracks, like a railroad track. I'm drawing it out of proportion. This is very small. Compared to these guys, this is very small. Am I making this is called, I'm talking about microtubules, okay? The first one, okay? So what happens, there are vesicles, which we talked about it. These vesicles can have either water in them or protein molecules, insulin. For example, an example of that is insulin. You all know about insulin. And what happens, these vesicles that are full of water or protein, the only way they get out of the cell is going through this railroad track. They move along and they get out. And that's another function of the cytoskeleton. That's another function of the cytoskeleton. They provide like a tracks, like a road for the vesicles to get out. And then whenever you take cell biology, they will talk about that in detail. Those of you who are going to go to biology. Those of you who are going to go to engineering, I guess you don't have to worry about it later on, you have to worry about it now for this class. But anyway, here you go. Here's a vesicle, here is the leg. Do not worry about all of that detail, what happens here. Here is a cytoskeleton, microtubules, and then little by little, they're moving out. The vesicles here is a transmission electron picture, cytoskeleton, vesicles, and of course the vesicle is moving out of the cell, whatever product it has, excess water or whatever. Here, again, you guys, listen, I don't expect you to know the numbers. Do not worry about the numbers, 25 nanometer, 15 nanometer, but the microtubule is the thickest one, microfilament is the thinnest one, which is actin found in your muscle cells, and then, of course, intermediate filament is the middle, as far as the size goes, is the middle. So there are three different types of cytoskeletons. And then, of course, there are functions in there. If you don't have to know, just the ones that I shared with you. Microtubules are hollow rod, about 25 nanometer diameters, and 200 nanometer to 25 microns long, usually. Functions of microtubules: shaping the cell, yes, guiding the movement of organelles, right here, and separating chromosomes during cell division, something we will talk about it later. But it's good to know a little bit of that information now. Here they are, microtubules, 9 platelets, 9 triplets. You have, if you look at it, there's nothing in the middle, and they have 1, 2, 3, 1, 2, 3, 1, 2, 3. There are 9 of them around, and you will learn about this a little bit later on, but just when the cells are going to separate, the cells divide, not separate. When you have one cell becomes 2 cells, these guys pull the chromosomes apart, the microtubules. And we will see that when we talk about cell division later on, exam material for exam number 3. But again, be familiar with it a little bit. I might ask you something. The combination of these two is called centrosome. Centrosome right here, and then of course chromosomes are inside of the nucleus, and they will pull them apart. Here we go, longitudinal sections of centrosome. What are similarities and differences between cilia and flagella? You all know you have cilia in your trachea. You all know what trachea is right here. You inhale, and when air goes inside, you have cilia to protect you from getting dust and trash into your lungs. So they bring all of that dust and trash into your throat, and you clean your throat into the bathroom, not on the floors of Delta College, I hope. Okay, so, and the only place that we human have flagella, I'm trying to relate this to human, that you would understand this a little bit more, but we have flagella all over. We have protista that have flagella. We have other organisms that have a flagella. But the only place that human have flagella is human sperm. Females do not have any flagella. They have cilia in their fallopian tube and uterus to push the egg down. They have cilia in their trachea, just like men. But the only place you find flagella is in sperm, in case of human. But again, flagella is all over in Mother Nature. All over, we have them all over. Okay, microtubules control the beating of flagella and cilia. Yes, the flagella and cilia move. Okay, they are inside microtubules containing extension that projects from some cells. Cilia and flagella differ in their beating patterns. A basal body that anchors the cilium or flagella, a motor protein called dynein, which drives the bending movement of the cilium and flagella. These are some of, here we go, if you would, the way the flagella moves, it's like this, okay? It moves up and down, but the cilia moves like a power stroke. They attach right to the surface of a cell membrane, and they go back and they beat like this. They call it recovery stroke and power stroke. And so the way the flagella and cilia move, they're different. Bottom line, you should know. If I ask an essay question, what are the differences between flagella and cilia? I didn't ask. I'm sorry, I should have asked that. Put on my list of what are the differences. And then the way flagella and cilia is, inside of, if you cut a flagella, this is a flagella, for example, you cut it like this, you cut it like that, and look through it this way, then you see 2 microtubules in the center and 9 microtubules which are in pair, 9 in the parameter. 9 in the parameter and 2 in the center. Brian, what is your question? Please ask. Don't worry.
I answered it.
You answered it. Ask him. How do you know? How do you know he gave you the right answer?
So we're just talking about it.
Huh? How do you know? I hope he gave you the right answer. Okay, so you have this, call this 9 plus 2. 9 plus 2 microtubule in the center, 9 in the parameter. Here's a longitudinal cut of a cilia or flagella. Here is a cilia. And then cross section of the basal body at the base. At the base, it just they have triplets, 1, 2, 3, just like the centrosome that I showed you. They do not have 2 in the center. They have just 9 in the parameters, and it's triplets. Here they are, animation. I might as well. Let's look at it. I forgot to ask you a question.
Flagella, or singular flagellum, and cilia, or singular cilium, are microtubular structures that extend outside some cells and that assist in moving the cell or the cell's surroundings. Flagella are much longer than cilia. When a cell has cilia, their number is large, whereas a cell will have few flagella, or a single flagellum. Many protists have cilia, and the sperm of many plants and animals have flagella. Flagella and cilia are assembled from tubulin subunits organized to form a circular arrangement of 9 pairs of microtubules attached to a central pair. Flagella and cilia bend to cause movement of either the cell or the surroundings. The bending uses energy derived from the hydrolysis of ATP.
Okay, something I forgot to mention. What did she say? Flagella are longer and cilia are shorter. That's another difference. Flagella are longer, cilia are shorter. The way they beat is different. They both need ATP for their beating. Those are some of the main differences. And flagella, there are a few of them. If an animal has flagella or an organism has flagella, you see 2, 3, 4, 5, 10 maximum. But cilia, you see zillions of them. That's another difference. The numbers of flagella on one organism is less, all the way from 1 to 10, 20, and cilia, zillions. You never see one cilia. If there was one cilia, then we'd call it flagella. Okay, oh, what happened? Intermediate filaments, is that right? So we did talk about cytoplasmic streaming is a circular flow of cytoplasm within cell. That is something you're gonna do, not today, you're gonna do maybe next week sometime. The streaming speeds distribution of materials within the cell. In plant cells, actin and myosin interaction and salt and gel transformation drive cytoplasmic stream. Okay, intermediate filaments quickly ranges from 8 to 12 nanometer, larger than micro filaments, but smaller than microtubules. They support cell shape and fix organelles in place. Intermediate filaments are more permanent cytoskeleton fixture than the other two. I guess I didn't talk about microfilaments. What are extracellular components? One of them is cell wall. Prokaryotes, fungi, and some unicellular eukaryotes have cell wall. The cell wall protects the plant cells, maintains its shape, and prevents excessive uptake of water. Cellulose fibers embedded in the polysaccharides and protein molecules. Any of that information about cell wall is new to you. I don't think any of that information is new to you. All of it you already know. Okay, so all of that information you already know. The extracellular matrix, ECM, of animal cells is made up of glycoproteins. I erased it already, but you all know what glycoproteins is, such as collagen, proteoglycans, and fibronectin. ECM, extracellular matrix, proteins bind to receptor proteins in the plasma membrane called integrins. I will talk about integrins in great detail later on, but here they are. Here is a cell membrane, phospholipid bilayer, protein molecules, cytoskeletons, right inside of the cell. And then outside of the cell, you have that structure they call it extracellular matrix. And they have a variety of functions. The cytoskeleton outside of the cell have a variety of functions. And then here is the glycoproteins and so on and so forth. Extracellular matrix, ECM, has a influential role in the lives of the cell, communicating with cells and activity of genes and nucleus chemical signals in the cell, a few functions. What is plasmodesmata in plant cells? A junction between two cells. Plasmodesmata are channels that perforate plant cells. Through plasmodesmata, water and small solutes and sometimes proteins and RNA can pass from cell to cell. So these are plasmodesmata right here between two cells. This is one cell, this is another cell. So molecules can go from one cell to the next cell. That's what it is, the junction between the two cells. I'm getting to the last question on your essay questions. Here they go again. Water in tight junction, desmosomes and gap junctions in animal cells. These are all in animal cells. 3 types of cell junctions. That's the last question on your essay question. This is the answer of the question in your last essay question. The only thing I forgot, you guys, is the difference between cilia and flagella, to put it in your essay questions. So everything else, it was in your essay question. I talked about it. You should know, except the differences between flagella and cilia, and what are they made up of, and everything. Explain structure of flagella, explain structure of cilia. Or was it in your questions? Was it in your list on your homework questions? Make sure you know that for exam purposes. But anyway, 3 types of junctions between the cells. In epithelial tissue, you have tight junctions preventing leakage of cell materials. So what happens? You have a cell. What a tight junction is, and I guess you will see some pictures as well. You will see some pictures. I'm not going to draw it. So that's between 2 epithelial cells, like your intestine. In your intestine, the 2 epithelial cells are they're tight together, so molecules will not go from here to here, from this cell to this cell. That's what a tight junction is. Desmosomes anchoring junctions fasten cells together in a strong sheet. So what desmosomes are, they attach cells to a base, to a ground, to a table, to a connective tissue. You guys don't know what connective tissue is. That's what desmosomes are. And gap junctions communicating. So like your heart cells, the cells in your heart, they're connected together. They're separate cells, but they have a little bit of junction between them. That junction is called gap junction, which we call it integrated disc when you get to zoology. Cytoplasmic channels between adjacent cells. Are you ready for some pictures? All those three, well, let's have some pictures. Here it is, the tight junction between this cell and that cell. You guys see that? Right here is a tight junction. Between that cell and that cell is a tight junction. Transmission electron picture. And then you have desmosomes. They are attaching, these are by filaments, they attach to the other cells. This cell is attaching to that cell. And gap junction is there are little holes, if you would, little channels that allow materials from this cell goes to that cell. They, by mistake, I think it's a mistake, they showed all of these three to you in one slide, but they all do not happen in one place. They all happen in different places of our body. Okay, here is an animation of desmosomes. Let's watch it.
Desmosomes, or anchoring junctions, appear as thickened patches in the cell membrane region between two cells. Desmosomes contain specialized proteins such as keratin, the same protein found in fingernails and hair, that increase the rigidity of tissues. Desmosomes, such as those found in epithelium, smooth muscle, and many other animal tissues, are button-like junctions that bind cells together and also function as anchors for fibers in the cytoskeleton.
Okay, I think there's another gap. This is gap junction.
In gap junctions, or communicating junctions, two cells are separated by a small gap, which is bridged by specialized channels that allow the passage of water and small molecules.
Big molecules cannot pass through.
Coordinate the activities of adjacent cells. For example, a hormone that stimulates one cell will often activate adjacent cells as well by the passage of intracellular signals through gap junctions.
She didn't mention heart, but that's okay. Oh, tight junction.
Tight junctions, which bind cells together, are found predominantly in epithelial tissues, such as the lining of the intestines. Tight junctions form a barrier that prevents fluids from moving between the intestinal space and the space between cells.
They cannot get in. So the epithelial cells in your intestine, molecules cannot get between them. Okay? Between them. I didn't say through them. The molecules will get through them. You have your epithelial cells like this, they have brush border. The molecules, the coffee you drank this morning or the food, the egg you had this morning, they go through them and they go to blood vessel here. But the molecule from here, from this cell, cannot get into that cell. That's what the tight junction is. Am I making some sense? These two are closely anchored to each other, so molecules from this cell cannot get to that cell. But the molecule from here is the coffee or orange juice, an egg you ate, it goes through, and it goes to blood vessel, and the blood vessel will carry it to your brain. Am I making some sense? But the coffee or the juice or whatever you drank this morning does not get from this cell to that cell. Am I making some sense? They're anchored together so tightly, so that's what this one is saying. The molecules cannot. But the other ones, like the cells in your heart, this one, they're connected together. So molecules go from this cell to that cell. That's called gap junction. Okay, so make sure you study those three differences. That's an essay question in your homework I asked. Okay, you guys take a 5 minutes break and then get up, go move around, and then we start chapter 7.